Today I'm going to be talking about transcription and translation and different mutations that can occur during this that affect those processes. So whenever you're trying to look for the amino acid for a certain protein, we're going to be reading a 5' to 3' direction to find that coding strand. And within that coding strand, we're going to start off by finding our start codon, which is ATG, which is underlined in blue, and our stop codons, which are underlined in red. It looks like the bottom strand is going to be our sequence of interest because it has 1, 2, 3, 4, 5, and this has 1, 2, 3, 4 amino acids. So taking our coding strand, we're going to write it in a 5' to 3' direction to make it easier. And after transcription, the coding strand is going to be the same as the mRNA, except for the thymines are going to be uracils. So we're going to place the T's with U's. And then after translation, using this our key right here, we can determine what amino acids are going to be encoded for in the chain. So we have AUG being methionine, GUC being valine, AGG being arginine, UUC being phenylalanine, AAG being lysine, and UAA being the stop codon, leaving us with our 5 amino acid chain. But mutations can occur, specifically point mutations that we're going to be talking about in this video, which are exchanges nucleotides, which can affect this the products of translation. A transition point mutation is when you change from a single nucleotide from a purine to another pyridine, or a pyrimidine to another pyridine. A transversion is when you change a purine to a pyrimidine, or a pyrimidine to a purine. Here we have our original coding strands, so we can compare. The first point mutation we're going to talk about is a missense mutation. In this example, we change this G to a C, which is a transversion. And here you can see a missense mutation occurs whenever the nucleotide exchange ends up with a different amino acid than before, meaning it used to be an arginine, but now it's a serine. Now looking at a silent mutation, it's different because when you do, for example, this cytosine to adenine, which is a transversion, you end up with the same amino acid because this originally was a valine. Now, looking at a nonsense mutation, you encode a stop codon instead of that normal amino acid that used to be. Right here, as you can see at this UAG, it encodes for stop, meaning a shorter amino acid chain. And a read-through mutation occurs whenever the stop codon is mutated, and for example, this adenine to cytosine, which is a transversion, and the ribosome is going to create a longer amino acid chain than there needs to be. And also this nonsense mutation is also a transversion.